Hey everyone, it's Mr. Wolfe here, and I'm going to be going over DNA replication in this video. Alright, so first of all, DNA replication is going to happen because cells need to duplicate their DNA to do mitosis for those cells to divide. And this is all going to take place in the nucleus of a cell. So I'm going to start off with my DNA molecule right here, and you'll notice that it's made out of a bunch of different types of nucleotides. I have two strands of DNA that are connected by these things called hydrogen bonds, and a lot of times in class we've been representing these as paper clips. I'm representing them as these blue dashed lines in between. I'm just showing that there's multiple ways to represent the same thing. All right, the first thing that's going to happen if we want to make a copy of DNA is an enzyme is going to come in and it's going to break these hydrogen bonds. And by breaking those hydrogen bonds, my DNA molecule starts to separate. All right, now these strands of DNA, they have a specific name, and the blue ones are going to be, or like the original DNA molecule, we're going to call these the parent strand. So this is a parent strand, this is a parent strand, because they came from that original DNA molecule. Now also in the nucleus of a cell, not only do you have these connected nucleotides in the DNA strand, but you also have these things called free nucleotides. And these are just nucleotides that are on their own floating around the nucleus. So what ends up happening is this parent strand right here, it's going to be used as a template to build another DNA strand. So you're going to have an enzyme that comes along, and it's going to grab the correct nucleotide, right? This one has a thymine, this one's an adenine, A and T's go together, and it's going to add them right here. And it's going to do the same thing for all these other ones. So we're going to have, where's my nucleotides? We're going to have this enzyme add the A with the T here. Enzyme 2 is going to add another, the G with a C right here. And then finally, a C with a G right here. Right? Now, as this enzyme is doing this, these hydrogen bonds are reforming within my DNA molecule. Now, for the purposes of time, I'm not going to model it, but the exact same thing would happen over here, where I would have the enzyme come by and add those free nucleotides over here as well. You know what? I'm just going to do this. So we would have enzyme 2 adding the nucleotides over here as well. So I'll shift this over, and those hydrogen bonds would also form right here. So now I have two identical DNA strands, right? Like AT right here, GC right here, GC. But if we look at the colors, I've made one blue and one purple for a reason. And that's because the blue one, like I said before, this is going to be the parent strand, right? Because it came from the original DNA molecule. And the new strand that was made by adding those free nucleotides, they're not free anymore, they're connected together. We're going to call this the daughter strand. So now I have two copies of DNA. This copy could go to one cell as it divides. This copy will go to the other cell as it divides.